Let's move on to lecture objective 2. We're going to explain in general terms how the polarity of epithelial cells enables them to absorb or secrete materials. So let's talk about the epithelial cells and their polarity. Now, when we think about the epithelia, let's just zoom out for a bit and talk about where we find epithelia and the purpose of epithelia. Epithelia is the lining tissues of our body. So we're going to find them anywhere that our body wants to have a barrier between the external environment. So our skin is an example of an epithelium. Our GI tract, the lumen, is an example of epithelia. Our blood vessels is an example of epithelium. Our reproductive tract, respiratory tract, any type of barrier between one tissue and the other is where we find epithelium. The second major category of where we see epithelial tissues is our glands, right? Our pancreas, liver, salivary glands, all of the glands of the body, reproductive glands like the ovaries and testes, all of those glandular tissues are also going to be made of epithelia. So epithelia is going to have a unique function because of its location in that it's going to need to transport fluid. It's also going to need to be involved in absorption and secretion across the surface of the epithelial cells. So if you look at an epithelial cell, for example, here's the apical surface of the cell facing the lumen of this tract. Let's say this is the gastrointestinal tract, for example. So the apical surface faces the lumen. This is where your food contents would be. The basolateral surface faces the basement membrane, which faces the underlying blood vessels and capillaries, okay? And then the cells are lined up one against the other to form these tight junctions. And the function of these junctions is to prevent any cellular material or any molecules from moving between the cells, as opposed to going through the apical surface and then through the basolateral surface, okay? So there are two membranes across which any molecules that need to be absorbed need to be able to pass to get into the blood. All right, so just reiterating, the tight junctions here are helping to prevent any paracellular transport, right? This is where we get into things like leaky gut syndrome. If you've ever heard of that term, this is where you have injury or damage. This is especially seen in folks who have gluten sensitivity, where gluten actually damages these junctions, and that inappropriately allows for particles to move in between those cells, right? This is why we refer to this as being a leaky gut, and that can cause a lot of inflammation and a lot of other GI symptoms as well. So these junctions need to be very tightly adhered to each other to prevent that type of paracellular transport. Let's talk about the structures on the various surfaces of these cells. So the apical surface will have leak channels. So sodium, for example, can leak right into the apical surface. The basolateral surface is going to have two features. It's going to have the sodium-potassium pump. This is what we looked at when we looked at the primary active transport. This is an example of that. The sodium-potassium pump using ATP to move sodium and potassium against their concentration gradients. The other type of pump that we see on the basolateral surface are potassium channels. So as you can see here, having both the pump channels and the leak channels helps us keep the relative concentration of sodium and potassium the same in our intracellular and extracellular fluid, respectively. So because potassium leaks across the membrane and gets pumped right back into the cell, sodium does the same thing. It leaks into the cell and gets pumped right back out. We never see any net accumulation or any net change of concentration of these ions in any of our fluid compartments. All right. The other interesting feature that we see on the basolateral surface is the glucose carriers. So glucose carriers exist on the basolateral surface, whereas the sodium-linked glucose transporter, this is an example of secondary active transport. This is where we take the energy of sodium leaking into the channel, leaking into the cell, excuse me, and we couple that energy and use that energy to move glucose against its concentration gradient. So we have a higher concentration of glucose inside the cell, a lower concentration of glucose in the lumen, right, as you're ingesting glucose and breaking that down. And so we want to move glucose against its concentration gradient. And so in order to do that, we need to get the energy by a secondary active transport. Now, in order to get that glucose through the basolateral surface and into our bloodstream, which is where we need it to be to circulate it, right? We need to move it passively from a high concentration to a low concentration. And this is where those GLUT4 transporters will come into play. So this is an example of facilitated diffusion on the basolateral surface. So notice how all three of the mechanisms of transport, all three of the carriers and transporters that we described in our second packet are coming back here on the exact same cell. So secondary active transport, primary active transport, and then passive transport facilitated diffusion, all happening on one cell membrane. Now, okay, so we just kind of described this, the movement of sodium, which is being coupled to the movement of glucose. Okay. Okay, so here's an interesting question just to recap this. In this scenario, where is the glucose concentration higher? Is it in the extracellular fluid or in the intracellular fluid? So where do we think that glucose concentration is going to be higher? Okay, so you're correct. This is going to be the intracellular fluid. Okay, the intracellular fluid. Okay, I'm seeing a few different responses, but the intracellular fluid is correct. So inside of the cell is where we're going to have the highest glucose concentration. And this makes sense because if you think about glucose moving passively out of the cell, but actively into the cell, this tells us that there's a lower concentration outside the cell here and a lower concentration outside the cell here, okay? So that glucose has an active gradient to get it into the cell and a passive gradient to get it out of the cell. Now, we can also think about the movement of water in terms of the solids that are being moved in order to affect the gradient in order to move water. So this is how our cells are going to secrete water or mucus, for example, right? Because mucus has a lot of water in it. So in order to do that, the first thing that happens is we pump solutes into the interstitial fluid. So this is the intracellular fluid, this is the extracellular or the lumen, and this is the interstitial fluid, the fluid that's around the tissues. So first, we pump solutes into the interstitial fluid because we're kind of trying to manufacture a gradient. We're trying to create a gradient in order to move that water in the direction that we want to move it. So because I want more water to end up here, I'm going to pump more solutes here, and that is going to exert the osmotic pull that we talked about earlier. So higher solutes are going to be here in the interstitial fluid. Lower solutes are going to be out here in the lumen. And so water is going to move passively right across the cell, right, especially in between here, and go right into the higher solute concentration because of the gradient that is created by the initial pumping of the solutes, okay? So this is how cells are able to absorb water from the lumen, right? When you drink water, this is how it moves across into your blood from the interstitial fluid, or excuse me, from the lumen into the interstitial fluid. Now, another example is the opposite direction of movement. This is how we think about, for example, mucus secretion. So both absorption and secretion relies on the movement of water. So a clinical relevance to this is cystic fibrosis. This is when patients have a genetic defect in the chloride transporters, so they cannot pump solutes into the lumen, right? We would initially need to create solute concentration in the lumen, and then water will simply follow. But in these patients with cystic fibrosis, there's a genetic defect in the carriers that allow us to pump this chloride out in order to make sodium chloride in the lumen. As a result, the mucus ends up being much thicker because it doesn't have enough water. This leads to various types of infections, especially lung infections. It also leads to infertility because the mucus in the reproductive tract is much thicker than it should be, much more viscous. And so this is a practical example of what can go wrong if water cannot move appropriately across our cell membranes. Okay. All right. The last thing here is just looking at transcytosis, so the movement of particles by cytotic vesicles, so endocytosis or exocytosis. And so basically we uptake these particles in a vesicle, move them across the membrane, and then release them on the opposite side.